All right, hi there, biology students. This is Miss Wythe coming at you with an activity that we did in class today called the mitosis card sort. So we have 7 cards, and they are going to be used to sort chronologically for the order of cell division or mitosis. These cards are one side, or these images are on one side. That's kind of like a computer drawing. And on the other side is a microscope image. So very different kind of image on one side. So the goal of mitosis is to go from one cell to 2. So our thinking process, we don't have any vocabulary. We don't know what we're looking at exactly, except we know we're looking at cells. This is one cell, right? I also see one cell, I see some others, and I see 2 over here. So I'm actually going to put the 2 here at the end. I'm going to put the 1 at the beginning over here. And, this one almost looks like 2. So I'm going to move that one towards the end. Okay. And like I said, this one kind of looks like that one. And then I got these 3 others. So I'm going to put these kind of in the middle for the time being. And let's talk about the beginning and the end of why I made some of these choices. So over here, we've got one cell again. You can see that the second one here, I made this decision to make this one the first one and this one the second one because this one's larger. So if a cell is going to divide, it's got to grow. So we can see some growth, right, in just a static picture here. You see this pokey thingy, if you look real close. Don't worry about the lines. They're actually labels of things that we've cut off out of the picture. But this pokey thingy, it looks like it doubled itself. So suddenly there's one of them and now there's 2 of them. I see 2 here, 2 pokey thingies, okay, so 2 pokey thingies. And as I look towards, I get to the end here, I've got 2 cells, which I've accomplished. That's pretty good. Here's the first one. So one cell ended up becoming 2. We're trying to figure out the whole process here. But each single cell has one of those pokey thingies. So we got that on the right track. This choice I made because, this is almost 2 cells. You can see it almost like an action pose of it just about to become 2. So we've got the beginning correct and we've got the end correct. What's going on with the middle here? So I can see a blue nucleus. It's weird looking nucleus over here and like back to normal over here. So this is that kind of DNA that we've talked about, the chromatin form or the messy spaghetti version of DNA. So I got to find a way to go from messy spaghetti DNA. All of these look a little different. This looks not like spaghetti. This is more that organized alphabet soup kind of DNA we've talked about, or the chromosome format, back to messy. So the ones that I'm looking here at the middle look to be part of the chromosome part of the process. So I'm going to spread out here, move these aside, and I've got a nucleus. This looks pretty close. Like jumping from here to here, from here to here seems pretty extreme. So this one I'm actually going to go with as my third. So if I look at the changes, these 2 pokey things, and I'll turn this one be on its side like that. So we doubled those, and now it looks like they're moving apart. Looks like the DNA in our nucleus went from that spread out chromatin form messy spaghetti to now like a getting organized. Not totally done. I still see some of the nucleus, but it looks like the outside border is breaking down maybe, right? And now these are my 2 choices that I have left over. And the end result is I see 1, 2, 3, 4 chromosomes here and 4 there. So right now I've got 4 X's and I got 8 individual pieces. So I'm going to make this decision here. So these look like really long, stringy X's of chromosomes, not quite condensed totally. And now here they look like they're fully condensed. They even look really organized, right? They're really lined up in the middle. This cell is like, really looks like it has its ducks in a row here. Everything's ready. And it looks like it's splitting everything. So if you look at the orange chromosome, it's being all in one piece, but here it got broken into 2. And here we've got an orange chromosome here and an orange chromosome there. If I look at the purple one, the second one, one of them broken into two pieces. Purple one here on the bottom, purple one on the top. Same thing with red and green, and so forth. If you look really closely without my shadows in line, in the drawing here, there's a whole lot of strings, but I see an orange, a purple, a red, and a blue. So it's like the chromosomes sort of uncondense themselves back into DNA in its chromatin form. So I'm going to zoom out here, try to get it all in view, right? So we've got one cell growing, doubling some of its stuff to get ready for cell division. DNA is still in chromatin form. Here we've got chromosomes, 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 back to chromatin form. So this raised part, these four in the middle here kind of, is the actual mitosis part. This is like getting ready. This is mitosis is over and you've accomplished the task. So we see the chromosome format of DNA here, chromatin format of DNA here. And if we look closely, you can see we've kind of condensing DNA, organized, lining it up in the middle, separating everything, packaging it back up in a nucleus, and get everything back in order the way it's supposed to be from the beginning. So this was just an activity. See, I've used hardly any vocabulary, but I can still just with pictures see what's going on. I'm going to now flip them over, and these are images from microscopes of the same thing that's happening. So let me flip them all, and then I'll do a quick go through of these, okay? So again, your nucleus kind of looks like a blob early on. Don't really see those pokey thingies, right? There's just one of them somewhere. The DNA is dyed blue, cell membrane stuff is going to be red, and proteins are going to be green, or it might look a little yellow on the camera color here. So we got two of those green protein pokey thingies, looking like there's some chromosomes in there, right? It's getting a little, it's messy, but we're changing the way the DNA looks. Here, still getting a little more spread out, but like I can see individual chromosomes, and it looks like the green pokey thingies spread out, and they're on opposite sides now. This was our DNA lined up in the center, so I'll flip that one over and kind of get an idea of what we're looking at there. I know it doesn't look organized to us, but that's really organized to the cell. Look how symmetrical everything is, and it's really getting ready to divide in half equally. And the next step is breaking it apart, right? So all those chromosomes that had a buddy just a moment ago, they left their buddy. And they said, see you later, going to opposite sides. So the green pokey thingies look like they're doing some pulling and stretching. And then now, what was just barely spread out really looks like it's getting packaged back up again. So the blue DNA is put into separate areas. The green is like pushing it apart now for sure. We almost can see two cells. And at the end of cell division, we have our two cells that look just like the original first one. Slightly different shapes. They're not perfect. All right. Hope this helps. See you later.